Oh hi! In our last video we made maltose, or at least we did on the drawing. I failed to find where the first and the fourth bonds were. But because this is a bond in a sugar between the fourth carbon in one glucose molecule and the first carbon in another one, it's called a 1,4 glycosidic bond. All right, and this is one of the reasons we have to be very, very clear on labeling the individual carbon atoms, so we're really clear on where the functional groups are that the enzymes will be able to put into their active sites and make happen. So what we did was we placed these together. We produced one oxygen here, and that produces a covalent bond with one carbon, the carbon number 1 from one glucose molecule and the carbon number 4 from another. And in doing that, we of course produced water. So let's have a look what I've gone on with on here. So here's a maltose molecule that we've made from joining two alpha glucose together, and here's another maltose which I just drew in between the videos. So we can also join maltose molecules together. So we put two monosaccharides together to make a disaccharide. A monosaccharide and a monosaccharide make a disaccharide. Alpha glucose plus alpha glucose equals maltose. And what we can also do is put two disaccharides together to make a polysaccharide. And so what we do is, oops, we haven't got that there yet, we take out two hydrogens and one oxygen, and we use the correct pen to do that, and we eventually end up with a condensation reaction which produces the water given off and therefore synthesizes a larger molecule from two smaller molecules. And if we continue that on, we get the molecule called amylose. Now, amylose is the kind of starch that's commonest in things like bread and rice and potatoes, and it's a single chain starch. There is a slightly more complex version of starch called amylopectin, which has branches which make 1,6 glycosidic bonds, and that is catalyzed by a different enzyme depending on exactly where the functional groups are, the OH groups are that will be involved in the reaction. But we can also do the reverse, and the reverse is called hydrolysis or hydrolysis. Hydro, of course, water, and lysis, destruction. And so in order to break these apart, we destroy the water. We break the water into the two H's and the O, and we have another enzyme, and this one's called amylase. Amylase, you've got in your saliva. You've got salivary amylase. You've also got pancreatic amylase, which goes through the pancreatic duct and into the duodenum, the first part of the small intestine. And that basically is involved in breaking up amylose into maltose. And so it reads the sequence of the atoms in these molecules, identifies the site at which it can cut between two maltose molecules, it takes in the functional groups into its active site, and then, with a little twist, as soon as it enters its active site, then it makes a small chemical change. And that small chemical change, of course, breaks a covalent bond. That oxygen is now stuck to this carbon, another oxygen is attached and tied in place by having their covalent bonds taken up with hydrogens. And now it's back to maltose, C12H22O11. All right, so now, obviously, there must be some sort of enzyme that can break maltose back up into alpha glucose. And not surprisingly, this is called maltase. So we do exactly the same thing. Here's the maltose made of the two rings of alpha glucose. The maltase takes it into its active site by recognizing its structure. As soon as this goes into its active site, that changes the enzyme's shape and it forces it to switch it over, break it apart, and, sorry about this, glad we're not doing this with anybody else in the room, and we're back to our 2-alpha glucose. And if that was not clear, which of course it wasn't, what I did for each of these was break that, add another oxygen to the one without the oxygen, and then reform the OH groups, the hydroxyl groups. The same thing can happen here with this maltose molecule being taken into the active site of a maltase enzyme. As soon as the molecule makes contact with the various parts of the active site, the enzyme molecule is shifted. That shift of the very large enzyme molecule breaks that, and now we can break up the water and use the water to remake our alpha glucose. So this one used the enzyme maltase. So maltase, well, here's what Amylase did. It took amylose and busted it up into maltose molecules, so OH. You notice it's quite easy to represent this without showing all of this guff here. This is far too complex, really, although you do need to know what it means. And then, of course, maltase breaks maltose into 2-alpha glucose molecules. So that's one of the basic parts of digestion. We don't really study human biology in the AP course. We do in the honours course, but you will need to know this part of it because it's so involved in the biochemistry.